Namaste. In this lecture, we will explore a basic but very important question. What is biodiversity? We often encounter this word in policy discussions, conservation documents, and public debate, but we do not always stop to define it carefully. Today, we will begin building a clear scientific understanding of biodiversity by looking at its forms and its levels of organization. Our path today is straightforward. We will first define biodiversity clearly and precisely. Then we will identify its main dimensions. After that, we will understand levels of biological organization, and we will use a real landscape example to make the idea more concrete. We will also correct some common misconceptions, and finally, we will connect today's topic to the larger course on sustainability. Let us begin with a simple question. When we say biodiversity, what do we really mean? Is it only about animals? Is it only about the number of species in a place? Or is it about variation throughout life in a much broader and deeper sense? These questions matter because the answer shapes how we study nature and how we make conservation and management decisions. A narrow sense will limit our thinking. A broader, more careful answer will open up the subject. Scientifically, biodiversity means the variety of life in all its forms and at all levels of organization. Biodiversity means the variety of life in all its forms and at all levels of organization. Now, this is a deliberately broad definition, and that breadth is very important. Biodiversity includes life that we can easily see, such as trees, birds, and mammals, but also life that is less visible or completely invisible to the naked eye. It also includes variation within living systems and variation among living systems, within and among. Biodiversity is therefore not just a list of organisms. It is the full range of living variation, from the genetic to the planetary scale. Let us unpack the phrase all its forms. What does it mean? Biodiversity includes plants, vertebrates, invertebrates, fungi, bacteria, and many other microorganisms, all different forms of life. This means that a biologically rich place is not rich only because it has large or attractive animals. It may also be rich because of its insect life, or its soil microbes, or its fungal networks, or its aquatic microorganisms. A complete understanding of biodiversity requires us to include all major forms of life, not just the most visible ones. Every group matters, even those that we cannot see without instruments, such as microscopes. And this leads us to an important distinction between visible and invisible biodiversity. Some biodiversity is easy to observe on a field visit. We can see a forest canopy. We can see birds in flight. We can see mammals grazing. But much biodiversity is microscopic. It is hidden in soil, hidden in water, hidden in roots, barks, and decaying matter. Hidden diversity can still shape major ecological processes. Nutrient cycling, decomposition, disease regulation, and soil formation all depend on organisms that we rarely see. And what is unseen can still be ecologically very essential. The phrase all levels of organization is equally important, if not more. Biodiversity exists at the level of genes, at the level of species, populations, communities, ecosystems, and even larger ecological systems. Now, this tells us that biodiversity is not located at only one scale. We can study variation within a single species, among many species, within a single habitat, or across a broad landscape. So we can look at it at different levels. A good scientific definition must therefore include these multiple levels because life is organized in layers that interact with one another. Each level adds something that the level below it cannot fully explain. At the heart of biodiversity is variation. The image on this slide shows birds flying in a V formation. Even within this single flock, each bird occupies a distinct position. Each bird expends a different amount of energy and responds slightly differently to the air around it. And that simple image captures something very important. What looks like a uniform group is actually made up of many small differences. And biodiversity works in a similar way. There is variation within species, meaning that individuals of the same species may differ in genes, they may differ in size, color, or behavior. There is variation among species, meaning different species can play different roles. There is variation among ecosystems, and there is variation in ecological roles. Without variation, biodiversity would be a much weaker scientific idea. Variation is what makes it strong. And from this broad definition, we usually describe several main dimensions of biodiversity. These are genetic diversity, species diversity, and ecosystem diversity. We may also include functional diversity as an applied extension. Functional diversity refers to the variety of roles that organisms play in a system. Think of a forest where some species provide shade, some pollinate flowers, some break down dead matter, and some keep other animal populations in check. Now, all of these are different functions, and each organism occupies a different niche, a different position within this living system. Two places may have similar species counts, and yet they may function very differently, especially if the key roles are missing. So, these dimensions together help us organize our thinking so that instead of using biodiversity as a vague word, we can precisely ask what kind of variation we are examining. Now, let us start with genetic diversity. Genetic diversity refers to variation within a species. It arises from differences in genes and in inherited traits. We do not usually see genetic diversity directly when observing nature, but we may see its effects. There may be differences of size, color, tolerance to heat, tolerance to drought, behavior, resistance to diseases, and so on. And many of these things are genetically controlled. So, genetic diversity is often hidden from casual observation, yet it is essential for the long term health of populations. It is the raw material of adaptation and evolutionary changes. So, why does genetic diversity matter so much? Well, it helps populations to adapt to changes. If all individuals within a species were genetically very similar, say clones of each other, then a single disease, drought, or environmental stress could affect them all in the same way. So there is no difference between them, so everybody would be affected in the same way. Greater genetic diversity reduces that vulnerability. So some individuals may carry traits that allow them to survive conditions that others cannot tolerate. And the surviving individuals can then re-establish the population when conditions return back to normalcy. Genetic diversity therefore matters for long-term population survival. When genetic diversity is lost, resilience may quietly but invisibly decline. The next familiar dimension is species diversity. This means the variety of species in a given system. It is often the dimension people think of first when they hear the word biodiversity. In a grassland, wetland, or forest, we may ask how many species are present and what kinds they are. Species diversity matters because different species occupy different niches, perform different functions, and contribute to the overall stability and productivity of the system. However, species diversity is important, but it is not the whole story, as we will now see. Here, we should pause and correct a very important and very common assumption. When people first hear the word biodiversity, they often think it simply means the number of species in a place. That number is called species richness. It's not called biodiversity, it's called species richness, the number of species in a place. And while it is an important, useful starting point, it is incomplete on its own. For instance, consider two forests, each with 50 species. In the first forest, one dominant tree makes up 90% of the vegetation, and most of the other species are rare. They comprise the remaining 10%. In the second, species are more evenly distributed and represent a wider range of ecological roles. So, by a simple count, both forests look equal, both have the same number of species, but ecologically, they are very different. Species identities matter because different species contribute differently to the overall system. Relative dominance also matters because a system that is heavily skewed towards one species is often more fragile. And a hidden diversity, such as soil microbes or invertebrates, may be missed entirely if we focus only on the visible counts. And so, species richness is a useful starting point, but it is not a complete measure of biodiversity. A third major dimension is ecosystem diversity. This refers to the variety of ecosystems and habitats across space. Different ecological systems support different life forms because temperature, moisture, soil type, nutrient availability, and disturbance patterns all vary from place to place. A coastline, a grassland, a wetland, and a tropical forest do not provide the same conditions and therefore do not support the same organisms. Biodiversity also includes these larger spatial differences. When we study biodiversity across a region, we are therefore also studying how many different kinds of living systems are present in that region. Functional diversity adds another important layer. It refers to the diversity of ecological roles and traits among organisms. Species do not all contribute to ecosystems in the same way. Some organisms pollinate, some decompose, some regulate prey populations, some fix atmospheric nitrogen, and some modify the physical habitat. Function matters alongside identity. Two systems may have similar species counts, similar distributions, and yet differ strongly in the ecological work that those species perform. And that is why functional diversity is especially useful in applied ecology and in conservation planning. We can now compare these four dimensions side by side. Genetic diversity is variation within a species. Species diversity is variation among species. Ecosystem diversity is variation among ecological systems. Functional diversity is variation in roles and in traits. Keeping these distinctions clear helps us avoid confusion. When we observe biodiversity changes, we should ask which dimension is changing, because the causes, consequences, and management responses may differ from one dimension to another. Now, let us move from dimensions of biodiversity to levels of ecological organization. The main levels are organism, population, community, ecosystem, biome, and biosphere. These levels help us see how life is structured from the smallest commonly studied ecological unit up to the planetary scale. As we move through these levels, our questions become broader and the patterns we look for become more and more complex. Each level is real, and each level provides different insights that we cannot get from any other level alone. The first level is the organism, meaning one individual living being. This is often the starting point for ecological observation. We may examine one tree, one bird, one insect, or one mammal. Individual organisms differ in their traits and performance. Some grow faster, survive longer, or respond differently to environmental stresses. Those individual differences can matter ecologically because they influence reproduction, competition, predation, and survival. So, even at the level of a single organism, variation is already present and already has consequences for the broader living system. The next level is the population, which means individuals of the same species living in the same area. A population is not simply a crowd of identical organisms. Populations contain genetic and trait variation, and their structure can differ from place to place. One population may be large and well connected, while another may be small and isolated. Biodiversity therefore includes within species structure. This is one reason why conserving a species name alone may not be sufficient if local populations that make it up are quietly disappearing. Building on that idea, a community includes multiple species living together in the same place. At this level, we begin to study coexistence and interaction. Communities reveal who lives together, who competes with whom, who eats whom, who pollinates whom, and how species affect each other's populations. Community composition can differ sharply even between places that look similar at the first glance. Two forests may have similar tree cover, yet support very different animal and plant communities beneath the canopy. Biodiversity therefore includes not only separate species counts, but also the particular combinations in which species occur together. The next level is the ecosystem. An ecosystem includes the living community together with its physical environment. This means we must consider both biotic and abiotic relationships. The living organisms matter, but so do abiotic things like water availability, soil composition, nutrient flows, light, and temperature. We ask not only what is present, but also how energy flows, how materials cycle, and how organisms interact with their surroundings. Ecosystem thinking expands biodiversity well beyond a list of organisms to include the processes that sustain life. Beyond the ecosystem level, we come to the biome. A biome is a large regional ecological zone defined mainly by climate and dominant vegetation type. The image on this slide shows a landscape from Manali in the western Himalayas, and it is a good example of a temperate coniferous forest biome. Here, cool temperatures, seasonal snowfall, and mountainous terrain together create conditions that support coniferous trees such as deodar and pine, along with the many organisms that depend on them. This is quite different from, say, a tropical forest or a desert, which have their own distinct climatic conditions and characteristic life forms. Different biomes support different biodiversity patterns, precisely because these large scale environmental conditions differ so strongly from one region to another. At the biome level, we are less focused on a single local site and more focused on these broad regional patterns of life. At the largest scale is the biosphere, which is the global sum of all life and all living systems on Earth. The image of Earth from space makes this point clearly. Local biodiversity is a part of a planetary whole. Every ecosystem, every population, and every organism contributes to the biosphere. Global changes such as climate change or widespread land conversion affect biodiversity at every level simultaneously, and losses of local biodiversity accumulate into planetary scale changes. Now, why do these levels of organization matter in practice? First, they prevent narrow thinking. If we focus on only one level, we may overlook important patterns at other levels. A species may persist in a database while its local populations decline and its functional role completely disappears. Second, these levels connect small scale and large scale processes. Changes in individual organisms can affect populations, which affect communities and ecosystems. Third, they help us conduct disciplined scientific study by defining where a pattern occurs and how it relates to patterns at other scales. Let us now use forest as a teaching example to bring these ideas together. A forest is much more than trees. It contains organisms at many levels and in many forms, and it includes both visible and hidden biodiversity. When we look carefully at a forest, we can observe genetic variation among individuals of the same species. Many different species of plants, animals, and microbes living together, communities interacting through competition and predation, and ecosystem processes unfolding within a defined physical environment. The forest is therefore a useful place to practice applying all the concepts that we have discussed. This slide adds an important practical point. In a forest, biodiversity may be detected directly or indirectly. The two images here illustrate this well. On one side, we see a tiger, a direct sighting of the animal itself. On the other, we see tiger pug marks, the footprints left behind in soft ground. In field ecology, we very often work with indirect signs like these rather than direct sightings because many animals are elusive, nocturnal, or simply rare. A pug mark can tell us that a tiger is present in that area, even if no one has seen the animal. And this principle also extends beyond tigers. Biodiversity in a forest may also be inferred from calls, nests, droppings, scratch marks on bark, or feeding signs. So, understanding and documenting biodiversity requires not just the ability to observe, but also the ability to read and interpret the signs that various organisms leave behind. If we ask what is actually in a forest, the answer must be broad. There are trees, shrubs, but also herbs and climbing plants. There are birds, mammals, reptiles, and insects. There are fungi, microbes, and decomposers working in the litter and soil layers. There is water in streams and soil, along with microhabitats and niches that many specialist organisms depend on. A niche refers to a role and position of an organism within its environment, including what it eats, where it lives, and how it interacts with others. This full view helps us appreciate the forest as a layered living system. This brings us to a common mistake worth correcting directly. Biodiversity is not only wild animals. Wild animals are certainly a part of biodiversity, but plants, fungi, microorganisms, and invertebrates matter just as much. In most ecosystems, these less celebrated groups perform basic ecological work, without which larger animals could not survive. Pollination, decomposition, soil formation, and nutrient cycling all depend on organisms that rarely appear in popular discussions. A narrow focus on large animals creates serious blind spots that can mislead both study and policy. A second common mistake is to reduce biodiversity to the species counts alone. Number matters, but not by itself. Composition matters because different species are not interchangeable. Genetic variation matters because it determines how well populations adapt over time. Ecological structure matters because relationships among species shape how the whole system behaves. And function can begin to change long before counts visibly decline. The food chain on this slide shows this clearly: grass, insect, frog, snake, and hawk. Pesticides enter the chain at the bottom, accumulating in insects and then in frogs, then in snakes. And by the time the hawk eats several snakes, the toxin has reached lethal levels. This process is called biomagnification. With hawks gone, snakes increase. So frogs decline, insects surge, and the grass cover falls. The species count has barely changed. We still have all the five species, and yet the system is already breaking down. An ecosystem may appear intact even when important losses are underway. The image on this slide shows a butterfly on a Lantana camara plant, photographed in Dehradun. To a casual observer, this looks like a healthy natural scene, a butterfly visiting a flowering plant. But Lantana camara is an invasive alien species, originally from Central America, that has spread aggressively across Indian forests and grasslands. It forms dense thickets that crowd out native vegetation, reducing the variety of plants that native insects, birds, and mammals depend on. The butterfly may still be present, the landscape may still look green, and yet the underlying biodiversity has already shifted. Native plant diversity has declined, and ecological relationships built around those native plants have weakened. This image is therefore a good reminder that appearance alone is not sufficient. We need deeper ecological understanding to judge whether biodiversity is actually being sustained or is quietly being replaced. Misunderstanding biodiversity is risky for several concrete reasons. It can lead to poor conservation priorities because we may protect only what is visible or popular. It can hide real ecological decline, especially when hidden diversity or functional change is not being measured. It can also oversimplify sustainability discussions by turning a complex scientific topic into mere slogans. Good policy and good management require a more careful understanding. Clear definitions affect what gets measured, what gets protected, and what gets lost. Now we can connect biodiversity to ecology. Ecology explains how living systems function. Biodiversity describes variation within those systems. Biodiversity without ecological context can become a static list, and ecology without attention to biodiversity can miss the importance of composition and variation. We need ecological thinking to understand why biodiversity matters, how it is maintained, and what happens when it changes. And this connection directly leads to sustainability. Biodiversity underpins ecological resilience, which is the ability of living systems to absorb disturbance and continue functioning. Human systems depend on living diversity in many ways, including food production, water regulation, soil formation, and climate stabilization. Sustainability therefore depends on more than visible abundance. A place may seem productive and healthy in the short term, while its underlying diversity is becoming simpler and more fragile. Diversity supports long term system stability, and that stability is what makes sustainable futures possible. And that is all for today. Thank you for your attention. Jai Hind.